So it's a pleasure to introduce the last speaker in this symposium. We've been getting Ron to visit here for a long, long time, and this is his first trip to Asia. So it's a major accomplishment to be able to get him to come here. So I think it really is a privilege to be able to listen to Ron and his ideas on how to change medicine, change society with the technology that's been developing. So as Dave indicated earlier, Ron of course developed some of the most important vectors that everybody used for the early molecular discoveries, including hepatitis C virus and all kinds of other viruses. Even earlier than that, Ron demonstrated how to use restriction enzymes to be able to join DNA together. So it was not thought before Ron's work that you could join two base overlap. Ron showed in very elegant work that using a restriction enzyme called EcoR1, you could join two pieces of DNA together. And even earlier than that, as a graduate student, he developed the most critical technology, electron microscope technology, that was used by both groups that discovered splice. So you can see from just that very short summary that from the beginning of his career, Ron has developed technologies that have changed the course of science, and he continues to do that to this day. However, what he's going to tell us about today are, Ron is now the director of a large center in Stanford, the Stanford Genome Technology Center, and he is now going to tell us how he's trying to change, in fact, medicine and society with the technology being developed. So it's a great privilege to welcome Ron to the Philippines, and I think we're in for a treat to listen to his ideas and changing. Ron Davis.
Well, thank you, Toto. Back in 1990, I set up a center referred to the Stanford Genome Technology Center with the sole purpose of really developing technologies. And that was a little unprecedented, but I was very fortunate that Jim Watson ran the Genome Project then and recognized the importance of developing technology. And our mission there was really not to apply technology, but in fact develop it. And what we found really was the best place to look to develop technology, not in a particular discipline, but in fact in between disciplines. So what we've learned to do is in fact team up people. We team up a student in molecular biology with an engineer, and we let them train each other in their discipline, and most of that is learning the language of the field. And they then start to look at a problem that they might deal with. Now, one way that they do that is they start putting together lists of problems that should be solved. And of course, there are thousands of them. Then they also look at what are the possible technologies that could be developed and try to find a pairing of this technology might solve that problem. And this takes months of effort and thinking. Once they have sort of identified an area of that type, then they go forward and start to develop it. And they develop it as a team. They may bring on new members of that team as needed. For example, some people are specialists in computer science or algorithm development and so forth. And so that's been sort of the style that we have done for almost every technology developed. Surprise to me is that it's not that hard. And when I say that, it means that what I'm trying to emphasize is I think a lot of people could do this. They think it's hard, so they don't. And it takes a lot of patience. And of course, a lot of funding agencies do not have that level of patience. And so the hardest part is to convince the funding agencies that you can do it. So when we write a proposal for our technologies, they're almost always turned down. And they're turned down as being they're totally impossible. This could not possibly be done. Our success has been because we have a big grant from the Genome Project that allows us to do the technologies even though they're impossible. And I think I'm tremendously benefited by having that. Now, so I'm just going to show you an area. We just picked out areas. They're not necessarily something that's the right thing to do. We just feel that we're in a good position to do it. So, as Toto mentioned, you look around you and take advantage of it. And that's what all scientists should do. Well, I'm at Stanford. So what is there? That's the hub of biotech in the world. It also has a fantastic nanofabrication facility. And so we should use it. So that was the first decision. We should involve local industry, and we should involve the nanofabrication facility at Stanford. That's our local resource. So we decided to just set up some parameters, design parameters we would call them. And what we wanted to do is to make some kind of very low-cost point-of-use device. Now, for a physician, that would be considered a point-of-care device. But for a biologist, it could be something in the field. And so we do not want to say we're going to target just medicine. We want to say this could be medicine, it could be for biological research, it could be for agricultural research. But what we want is something that's low cost and it can be used anywhere. And so these are just some of the design parameters that we put in place to say, okay, this is what it has to have. It has to be able to do these kinds of things if you're going to make a device that will give us information. Now, how to possibly use these, and again, we're putting some constraints on it. This is largely me. I said, okay, this is what we're going to do. I do not like lasers. We're not going to use any lasers, and we're not going to use any kind of dyes. Not that there's anything wrong with them, it's just that that's not what we're going to do. And in fact, we're going to use electricity. And so everything has to be read out as electrical signal or magnetic signal, but no colors. And that was just totally arbitrary on my part. And this was to try to make us more creative. This is not normally done. So this is forcing us to be creative. So when I started the Genome Center in 1990, I said, no radioactivity. All my students had a fit. How can you do research in molecular biology without using radioactivity? I said, figure it out. So at first they switched to dyes and lasers, and everything was done by fluorescence. So I decided, okay, now it's time to get rid of them. Go to the next thing. If you can get an electrical signal, very easy to amplify it. It's also very cheap. There's no reagents, no expensive compounds. But one of the areas is to really focus on trying to keep the cost low. That's important for medicine, but it's also very important for biological research. The major probably restriction that you have in doing any of your research is that it's too expensive. We've got to make it very cheap. So I'm just going to show you some examples of things that have been done that work, that help to address this sort of an issue, and then take it further where that where you can take that. Now, with these constraints and using electrical and magnetic detection, where what we're doing is really fusing biology and effectively solid-state physics. That's how we're going to get our electrical signals or our magnetic signals. Now, this interface here is not at all well understood. That's that area that is the void. Nobody works in that area. There is no expert. There is no discipline. There's no textbooks. So we have to figure it out. So this is the area right in here that we all work in. Now, because it's a solid-state physics and that we're at Stanford, this is all going to be done at the nanoscale, and this is all hopefully will help us keep the cost down. Now, we're really state-of-the-art there in our nanoscale fabrication. We're about 18 nanometers in our ability to fabricate. So this computer is 35 nanometers. So we're well below the state-of-the-art of Intel making computers. So we get things extremely small. So here's an example of one. This was developed in 2005. It was a job for an electrical engineering student who then put this together. And the idea was to use a magnetic particle for detection. So maybe if you're in biology, you've heard of the ELISA assay. And it's an assay that takes an antibody with this shape of this Y and is attached to a surface. It then reacts with something, a protein or some other molecule, and now that makes this molecule stuck to the surface because its antibody is stuck to the surface. And then you can bring in a second antibody, which will improve your specificity. And that antibody then will make what's called a sandwich assay. And what is typically happens in the normal circumstances, you put a fluorescent group there, and it now makes it fluoresce. Or you put an enzyme on, and you have an enzyme activity. But instead of doing that, we simply attach a magnetic particle. Now, there's several advantages of that. One, these things are unbelievably cheap. They're just iron oxide. And they're very stable. So this was a development with IBM to make these tiny magnetic particles. They're now commercially available. We buy them. But another real advantage of this is that by using magnetic instead of light, that the magnetic field, in order to detect it, you have to be very close to its source. And it falls off very fast in distance. So what do we use down here to detect that magnetic particle is the same, it's called the spin valve. It's what used to be used in hard drives. They use a different device today. But it's a very mature, advanced technology for detecting magnetic fields, which is how you write on your computer hard drive. So we're just going to be borrowing that from that industry. Now these are actually quite small, and you can make them very small detectors. And so in fact, it's very easy to make a large array of these detectors. So that allows you to make many, many types of tests. And we in fact set up a company once we developed this called MagArray, which is now using it to do commercial development. Now, if this nanoparticle, magnetic particle, is out in solution, it's not seen. It's too far away. Or if it's moving, it's not seen because it has to be stable in order to see it. So that means that when you do this assay, you don't have to do any washing. And it also means it's real time. So you don't go to an endpoint assay, you simply throw it in and let it in, and you can see it in functional time. Because these are very small, you can do like 1,000 different assays on one device, or in fact, we could probably set up the footage so that you can in fact do sequentially one assay. You only want to do one assay, but your one instrument can probably be used 1,000 or 10,000 times. That greatly reduces your cost. Here's just an example of, this is the very first reaction that was run on the device. And you can see there's buffer here. We pour off the buffer, you get an air interface, and that affects the magnetic detection. And then we add the reagents that we want to, the sample that we want to detect. And what you can see is this is a signal over here going up you can see the signal goes up pretty rapidly. And this is in real time. And this is in minutes. So you can see from this that in a few minutes, you can get a pretty good idea of the concentration of whatever you've had. That could be quite important for medicine if you need a fast assay. Instead of sending it off the lab and waiting an hour for them to run a standard ELISA and get it back to you electronically, or in fact, not get it back to you that day, but get it back to you the next day. If this was a bedside device, you would know the answer in 5 minutes. If you're in agriculture and you want to look for aflatoxins in some of the food source, we developed this for aflatoxin as well. You would know that this sample of food has aflatoxin in it and roughly how much it has in a matter of minutes. So that's sort of the style. Taking technology and develop it for biology. The whole problem with this thing was how to treat the magnetic detector so we could put the biological molecules on top of it. And the problem is, what are the metals that we have to work with? We have to deal with silicon oxide, gold, aluminum, chromium, titanium oxide. Those are the surfaces that we have to work with in nanofabrication. None of them are very good for biology. And so often we have to, in fact, make different layers of things to try to go from the solid state physics to the biology. Here's another example. This was developed in 2002. The idea is, could we detect DNA synthesis electronically by an electrical signal? What was done was to make a nanofabricated surface, put a gold plate on it, and then have molecules onto the gold that would prevent the gold from interfering with the subsequent biology. And then attach the DNA molecule to it. And that's here. You then put a complement molecule on it and make a double-stranded molecule. And this is now a substrate for the enzyme DNA polymerase. What happens when the DNA polymerase is here and you give it its bases as triphosphates to incorporate, it will incorporate a base. Now, how you use it for sequencing means that you only put in one of them, and now will it go in? And you look for a signal. So in this case, it's a G, and so if you use CTP here, then we would put that CTP, we put that C in, and we want to be able to see that signal. What happens when that base is put in is that it fixes a negative charge, because when you put in a new base, you have a phosphate there, and that phosphate is fixed. So it moves around normally, and all of a sudden it gets fixed. That causes a perturbation in the electric field around the electrode. What also happens is the hydrogen ion is created by the chemistry, and that hydrogen ion diffuses very fast. So there's two ways you can do this. You can either attach the DNA to the surface, and as this hydrogen ion diffuses away rapidly, you can get capture the transient negative charge that's put there, or you don't attach it to the surface. You put it on a bead above it, well above it, so that when you put the base in, the hydrogen ion diffuses away rapidly, and that's detected. And you detect the positive charge from the hydrogen. Effectively, it's a pH detection. We had a very difficult time commercializing this. Nobody wanted it. They were perfectly happy using gels. Why would you want to do DNA sequencing electrically? And it wasn't until a small company called Ion Torrent became interested in this that licensed the technology. But that's one of the problems we do have. How do we export something that sat around for a very long time before it was exported? This is the old guy, David Corman. But this is now a commercial instrument. This is what the instrument looks like. It's fairly small, sort of like this. And it's called a personal genome machine. It will do fairly high throughput sequencing, but it's possibly very geared towards smaller laboratories that don't have a huge infrastructure. The instrument, I don't know exactly what it's going to sell for. With time, roughly $50,000. One run on this instrument costs about $50, which is the cost of this CMOS chip that goes into it. I mean, that's what they charge for. Let me show you one other little technology that we developed to look at molecules. And this is also done on a gold surface. But this is trying to be able to do something label-free. I said I don't need lasers. And so this is an attempt to see DNA by excipients. And what happens is that we have a gold surface, we put DNA molecules here, short ones. And the goal here was really to look at these very short RNA molecules for clinical. So these are molecules that are around 20 bases long. They're very hard to work with because they're so short. People try to amplify them by sticking other sequences onto them and then amplifying, but you get a quantitation that goes to 5. And so we decided to try to detect them without any label, without any amplification. And so they are hybridized, and they hybridize their chip, and you can simply scan this with a force field microscope chip. And these little spots that you can see here are the hybridization reactions. So you can directly see the hybridization. We've now set up a collaboration with a small company that does electron optics. And he feels that on this gold surface, we can bounce electrons off this surface and be able to also see the DNA. And hopefully it'll be such high contrast that we'll actually be able to sequence the DNA directly. So that's another experiment to try to do. Now this idea was scan this was an engineering student came to me with his idea, and then we simply implemented the chemistry that we already had. Now I'm going to show you another device. This one is it's currently under development, and this is an attempt to maybe replace that instrument that I showed you for ion touring. And can we do better? So that ion touring instrument uses a CMOS chip. Pretty cheap to make them. They're $20 or so, maybe $50. But it's disposable. And so you could say, well, that's a lot cheaper than current technology. That's true, but you're still throwing away something every time. You have to throw it away because it has little wells in it that captures the beads, and you can't get the beads out. So the whole design is, and they're happy because they want to throw it away because they want to sell you another one. So could we design something that was reusable and with more sensitivity? So this is the device. It's a coaxial nanowire, and this can be used either for protein assays or for a DNA sequence. For a protein assay, what we do is to attach antibodies to the tip of this. This is a wire that's about 20 nanometers. So it's a little needle about 20 nanometers in size, which we then can put antibodies on. And if we react it to like an antigen, a protein, that in fact will perturb the electric field between these two conductors. And you can store it as a positive hit. The other way to use it as a DNA sequencer is just like before. That is, we can see the electrical reaction here, the hydrogen ion, the pH, the pH effect. But now it's a little wire sticking up off the surface. We have to then get a bead to be up here. Now that has tremendous advantages if we can put a bead up there, because now we can wash it. So the problem with putting something in a well, you can't wash it. If you can't wash it, you can't get efficient reactions all the time. And that's why it has a bead link. If we put the bead up here, we can wash it very, very well. But how do you put it up there? Well, what we're going to do is put another plate here, and we fabricate small magnets. Now these magnets are around 20 nanometers. We've already done all this. And we put an array of magnets and then use a magnetic bead, and we can capture the bead with these magnets. In fact, we can do it very unbelievably efficient. Every magnet pair will capture a bead. We have no empty spaces, and no spaces have two beads. So it's extremely efficient. And that then puts the bead right above the detector. But the nice thing about this is that we work out conditions that we can now strip the beads away from the magnets and reuse them. So it's our guess that we should be able to use this 100, maybe 1,000 times in reuse mode. Now normally what people worry about is contamination, right? You throw things away because you don't want to wash them. Well, but if you put the washing as part of the instrument, which is what this is going to be, that is, we now can redevise how we are going to clean the device, and now it can be reused. And it's just part of the instrumentation design, to learn how to get rid of what is already there. And there's a lot of tricks to that. So we're optimistic that this may actually give us a high-sensitive device. I'm going to show you another device. And this is unbelievably simple, but I think it's going to revolutionize diagnostic medicine because it's simple. So this is a fusion of a graduate student, a postdoctoral fellow in biology who worked on kinase. Okay, so whatever we do is going to be on a kinase because you already know the field. And an electrical engineer. So she had an antibody to kinase, the products of the kinase reaction. So kinases take a peptide and they put a phosphate group on it. Very important for cancer and all sorts of other biological processes. So you want to be able to detect that phosphate being put there. What is normally done is use an antibody to detect that, and that's the standard ELISA assay that everybody uses. So this is the antibody here that will react with this phosphate. But instead of putting a fluorescent group here or an enzyme, we simply put a bead. Now that bead can be it could be an organic bead, it could be a gold bead. Right? But a simple bead. Now when we put a plate here that has all these substrates on it will react, and then we put in the antibody that will detect it on this little surface. We use about a microliter. We can do this in smaller amounts. Now what we do is we simply pull off all of these beads by flow, and then put a little counter here, which is just a capacitance counter, and as the bead comes past, it clicks it off and you count it. So we've gone from trying to quantitate things to actually counting molecular reactions, and it's really trivial. So each bead is a single antibody reaction. Now, there's some tricks to this, but I'm going to show you the sensitivity of this. So we've just detected how sensitive a reaction can be. And right now, we are being able to detect 10 phosphate groups is our detection system at the present time. And so this has very, very high sensitivity. And when we compared it to the standard ELISA assay that was used in this, it's roughly 100,000 times more sensitive. So if you start to ask yourself, I now have an assay that is 100,000 times more sensitive than the previous assays. It's hard to use your brain to know what you can do with it, because that's a big factor. So one of the things we're going to try to do is to use it to test for TB. So one of the problems in Africa is a lot of children have TB. Really tough to do the sputum assay on them. This is just coming from the physician in Africa. They've asked us could you do something like this? Our guess is that there'll be enough small molecules coming from the TB that will be in the aerosols from the breath that we can simply take a device, have them blow into it, and run a TB test. We don't have to have the organism. And that will be very cheap, and maybe we can build it so it can be useful as well. Now, here's one of the molecular tricks. Why does this work so well? And the electrical engineer figured this out from his experience. Now, normally, you just do the reaction and look at it. Well, that has a big background. There's a lot of things that are false signals, and that limits your detection sensitivity. But what he decided to do is to look at These are just the controls and ordinals. Here's the reaction itself. What flow rate do I have to run across the surface to pull the beads up? Because I want to test them later. And found, in fact, that it's a very sharp flow rate to get the beads up. If I'm below that, they don't come off. So you simply use a flow rate that's below that level, and it strips off all the low binding constant stuff. All the gratuitous signals get stripped away. And then you raise the flow rate in this region, and it'll pull off the specific reactions. In other words, the flow rate is proportional to the binding constant. And if you have a lower binding constant, you can strip it off before the specific one. You don't go up to very high flow rates because there may be something else gratuitously stuck there that has a higher binding constant than what you want. So this gives you a way to make a cut. This is for chemistry purposes. So this is a chemistry reaction. I have a binding constant. I only want the molecules with the right specific binding constant. And that helps to get rid of your noise and your background. That's why it's so sensitive. And after we're done, we then put a high flow rate through it to strip everything else off, and we can reuse it. Now, there's another benefit of this. We have sensitivity that's way beyond probably what we need for most things. How can we use that? Well, we can in fact convert high sensitivity into rapid detection. In other words, everybody tries to go to an endpoint, maximum reaction, and that's because they don't have much sensitivity. They have to. And sometimes that takes you 2 hours to get the endpoint. We have sensitivity to burn. So what if we just let the reaction run till it's 1% reactive, 0.1% reactive, and then test it? For example, in many of these, the second order reaction is proportional concentration. So you can get the concentration just by the kinetics of the rate of reaction. To test this, the engineer did it for a DNA hybridization. Now, normally DNA hybridization is overnight because DNA doesn't hybridize quickly. It has a slow rate. But by pushing up the sensitivity, he was able to show that in fact he could quantitate DNA hybridization detection in 5 seconds. Now, it's only a few molecules, doesn't matter, it's specific. Now, there's another device that has been built, and it hasn't been popular, and I'm just going to show you anyway. And I think it has to do with the name. It's called the nanopipette. Okay? And it's very simple to make. Anybody can make these. You simply pull out a glass tube. And you can buy little devices that do this. And you can pull a piece of glass out until the tip of this glass is roughly a hole of about 40 nanometers. Now, if you did that by fabrication methodology, you're talking about a billion-dollar facility. All you have to do is pull a piece of glass out, and you have a tip that has a roughly 40 nanometer hole. And you can measure things going through this, like little beads. You can derivatize the surfaces of it by putting antibodies there and detection, and having it react and detect things. And what you can measure is the electrical flow out of the nanopipette. So you can detect and count these molecules. You can actually quantitate these. You can actually use sort of combinations of these and detect them. They're incredibly cheap to make, and they're very small. You can penetrate a cell with them. You can measure in vivo concentrations of proteins with it. They're small enough that you can penetrate an organelle. You can penetrate the mitochondria or the nucleus, and you can measure concentration. The nature of the, and probably the purity of this, when we use antibodies to do detection, those antibodies have an off rate. And so, in fact, again, you can wash it and reuse it. So it's not only very cheap to make, but you don't even have to throw it away. And we think that we can figure out a good application of this. It's so simple that it should be usable. But I think people are attracted by it because of the name pipette. And I keep telling them the student that was working in this come up with another name. Well, we do have another high-tech technology development. I don't know that this will work. We're really pushing the limit. Theoretically, it will work. The question is, can we build it? And it's real hard. So it's a nanopore, and nanopores have been around for a number of purposes for detecting things. This is a very different type of nanopore. We're trying to make it into a charge sensor and being able to see a single charge. Now, this is all done theoretically, and we found theoretically that we could use a fairly large hole and bring DNA through it, and that the negative charges on the DNA are normally coated with positive charges, sodium or potassium or whatever you use for the counterion. And that prevents you from seeing the phosphates on the DNA because they're shielded. But we put a very large voltage drop across here, not because there's a huge voltage itself, but because this is so thin. This is going to be down, I think we got it down now to about 50 nanometers. So all the voltage drop goes across 50 nanometers. So the voltage gradient here is something like 1,000,000 volts per centimeter or something. So it's a huge, huge voltage drop. And that strips away all the counterions. And that allows us now to see charges. And the technical problem is, so this theoretically will work. The problem is we have to build an amplifier. And the amplifier has to be within nanometers of this hole. And the problem is the technology we use to build the hole and build these wires is fully incompatible with the technology to make an amplifier. So we have to somehow rig this or change the technology to get this amplifier so we're really close. Now what can we do with it? It's going to be very cheap to build if we can figure this problem out. But if you put DNA through this, you can simply count the number of charges. You can measure length. This goes back to electrophoresis days. But it will have no length limit. You simply count the charges. So you'll reintroduce people to being able to size molecules, which is what forensics is really a sizing thing. This would do a good job for forensics. But in addition to that, we can put charges on the bases, and we can use it as a DNA sequencer. And this would make DNA sequencing unbelievably cheap because there's basically no reagents, and this device is probably reusable. And this is all being done at the 18 nanometer fabrication facility. Now, one of the things we did find with this, discovered with it when we were building it, is that when we put a small electric field across here, the DNA should be pulled in, but it is not. And for some reason, and we haven't figured out the reason, this is somehow gated. So DNA will not enter it. And you get to a critical threshold voltage and it will go in. And then when it goes in, it will go all the way through. So this has made the engineer that's doing this realize that with that principle, he can actually make a DNA transistor. And his game now is to try to figure out how to make a DNA transistor that's sequence compatible. So those are the kinds of technologies that you try to develop, and you use one idea to bootstrap you into another. Now, I want to go back to that size thing, because I think for the biologist, they lose track of size. When I say 18 nanometers, what is that? It's really small. So I just put up something that most biologists would know. Here's a human hair in cross-section for sizing standard. And here are the sizes of the instruments that we're building. Here's the sequencing instrument, that little dot there, and here's this multiplex protein analyzer there. All right? So these instruments are so small that you can't see them. In fact, they're so small you can't even see them in the light microscope. So that opens up a lot of possibilities. So the next thing that we're looking at now is working with some other individuals in electrical engineering to how to get signals out of these devices without having wires in them. And so there's a collaboration now to use radio waves to transmit information. If we can do that, then we can implant these in individuals. And so that means you might be able to put something in the bloodstream and have it do constant reading of biological information. And probably not interfere very much because it's much smaller than a cell. We could even implant it inside of the cell. So that's sort of a new way. I'm not sure anybody here is old enough to be a Star Trek fan. But in Star Trek, they always had this tricorder, which they took and waved over the person and feel out what they're wrong with them. And how in the world could you do that? Well, here's the solution. You implant something in the individuals that will transmit a signal, and then you can read it outside. Let me just give you a little brief discussions of some of the sequencing things. The genome center here, so I wanted to cover a little bit about sequencing. There's a lot of talk now with the new sequencers that you can get a lot of sequence. And a lot of people are talking about the complete human genome sequence. I call it a lot of hype because they don't really know what they have and they don't really know how to use it. But there's a lot of activity and talks and symposiums about it. But in fact, if you were interested in sort of clinical information, this is great for discovery, but I'm not sure it's very useful for clinical settings. And for clinical, the information that you want is the information that you know what to do with it. It doesn't do you much good to find that you have a base change here, but I have no idea what it means for a patient. And I don't see the reason even to spend the money to collect that information. Now, what a lot of people are saying is that it's going to get so cheap, why wouldn't you do it anyway? I would argue that it'll even be cheaper if they don't do it. And you're really always trying to reduce the cost. And so I think we're going to be talking about subsets. And I think it's mostly people aren't talking about this because they don't know how to do it. I'm going to skip these next two slides which show you how people are currently doing it. I don't think that's useful because it's going to go away. They cost about $1,000. But here are some of the ways that we do. How do you get a subset of the information instead of doing it all? So it's easy to do it all because you just take the DNA, randomly start sequencing, and then keep sequencing until you get it all covered. But I want a certain set of sequences out to do, and I want them to be limited, and I want only those to be looked at. Now, you normally would say you would do PCR. Well, that doesn't work because you can do for one. But if you start mixing PCRs, it doesn't work. Unless you do a lot of individual PCRs, then mix them together. But how do you do this multiplex amplification and selection? So we have two ways. We've actually developed about 5 different ways, but these are the ones that are currently being most actively pursued. We build an oligonucleotide, and then on its ends have the sequences that we want to go after in between. And the sequence that we want is here. But we don't actually target that sequence because the problem is in human there's a lot of pseudogenes. And if we target the gene that we're interested in, we'll wind up pulling out all the pseudogenes. And then you'll sequence those too, and then you have to use bioinformatics to figure out whether this is a real gene or it's a pseudogene, and it isn't very long, and I'm not sure if it is or not, which is what happens. But you go outside to a unique sequence to get rid of the problems of pseudogenes. There's only one sequence in the genome for here and one in there. You then hybridize this, so it hybridizes on both ends, and then use DNA polymerase to fill it in. And now you've captured the sequence. And then this ring, the circle, is easily amplified either by rolling circle or by PCR. And you can use common primers out here to do the amplification. And this works in high multiplex. So the highest we've gone so far is around 10,000. So we can do 10,000 of these simultaneously. We get about 98% of them working, and we don't lose quantitation. So if there's a deletion in the genome, that's preserved. It's going to be found there at half the concentration. There's another method that's not yet published, and this is the Illumina sequencer. It has a plate which you capture sequences on, and you do it at random. And there's sequence on that plate for doing the capturing. Instead of doing, and that captures everything randomly. But instead of doing that and trying to capture everything, we adapt their plate by putting another sequence onto that has the sequences that we want to capture. And if you don't know anything about the Illumina sequencer, this is going to make a lot of sense to you, and don't worry about it. All it is we advance the plate to capture just exactly what we want and use their plate to do all the capturing. And that allows us to sequence only the things that we want directly on the instrument. The cost of the other methods that people have been using is about $1,000. This is going to cost us somewhere between $1 and $30 to do this. So we just have a few of these targeted projects, just mostly test cases, but some of these are hard. So one of them is HLA. That's a very important region in the genome. It's one of the most diverse regions of the genome. In human genetics terms, it's a locus that an awful lot of genetic mappings go to. Many of the neurological diseases have a locus there. Autoimmune has a locus there, and so forth. We don't know much about it because people can't sequence it. And they can't sequence it because it's extremely high GC, which makes all of the reactions not work. And it's very polymorphic. And there's a lot of pseudogenes. But also cancer, we can do maybe 100 genes falling out that will help us diagnostic in cancer. We're looking at a lot of mitochondrial diseases. So we've identified about 1,000 nuclear encoded genes that are proteins that are in the mitochondria, and we're just going to sequence them. And we also do look at the immune system and survey that. I'm going to skip through this fairly quickly because I think it's pretty obvious. So we just heard about looking for the viruses. That's becoming relatively routine, where you can take blood and plasma, and it may have DNA in it. It may have virus particles in it, and you can DNA to kill all the cellular DNA and disrupt the virus and sequence it. Or you can take cells here and disrupt them again, pull out any protected DNA as a virus and sequence. This is routine. We do a lot of this in the lab. We do find new viruses. You can also do this on biological reagents. Are they contaminated? So we had it as a course to take all the vaccines that were around and sequence them. And in one case, a live attenuated rotavirus vaccine that was being used in the United States, half of it was a pig virus, a live pig virus. So we notified the CDC and the FDA, and they pulled that vaccine off. All the stocks were contaminated. They had no knowledge of that because how do you tell? And you do now. So now DNA sequencing is going to be a requirement for all vaccines. I think I'm running a little late in time, so I'm going to skip through some of this. We can now sequence HLA. It's working well. We're setting up to try to transfer that clinically. And we have a big contract now to do that on a lot of individuals. It's just a hard thing to do. But that's just the HLA locus, and I'm going to skip to the immune repertoire until I get to one other item that I wanted to talk about. And that's the future of drugs and also as molecular probes. And the reason to put this in is that we've been getting a lot of pushback from the drug industry about saying this can't be done and that can't be done, and every time they're wrong. And the problem is they have a lot of power, and it's expensive to show that they're wrong, and so their dogma just gets established and everybody believes it. And so we've been doing a number of tests about drug development. And this is a real simple experiment. So I just want to show you a few examples of this. So here's a case where we work with yeast. We like it because it's a eukaryotic organism and it's diploid. So it has 2 copies of everything. That's standard. That's just like any other higher organism. But we can delete a gene and identify, or we can delete both copies. So if it's in a gene that's essential, we cannot delete both copies. We can only delete one. And that is the way to identify in yeast what genes are essential. It turns out that there are only about 1,000 genes that are essential out of the 6,000. Only about 20% of the genes are essential. That prompted some people to say, well, gee, that means you can delete the other 5,000. No, it does not mean that. But what we're then trying to do is to say, okay, I can delete one copy of the gene. But now, and the organism is going to be fine. But if I put a small molecule in, that effectively decreases the copy. It's not really doing that, it's decreasing the amount of protein that's there. So if this can produce half the amount of protein as that one. But if I put a small molecule in, it can reduce the active protein even further. Now, if I simply put in a certain concentration of that, this cell will be more sensitive to that small molecule than to have two copies. So it allows you to identify the target. So what we've done is simply go through this at 6,000 genes. We've deleted every one of those genes. We look at them. We have a set of 6,000 deletions that have one copy. But you can go more than that. You can take and delete both copies. These are the non-essential genes. And in fact, you can look for other small molecules that will make this deletion important for the survival of the cell. This is intellectually a little hard to think about, because everybody likes to think of the drug directly binding to the product. The product's not there. It's not binding to the product. It's not there. But it's simply asking, can I find a small molecule that will make this cell, which seems to be going just fine without the gene, it will make it essential. And you can do one more thing. And then you can put in extra copies. And this, if there is a target then to the gene product of this particular cell will be more resistant to that small molecule. So you now have 3 different assays that you can run. What we do then is embed in each of these cells a DNA sequence code that allows us to identify it. 20 base pair sequence, that's why we make these things. We put this code in, which then means that all we have to do is mix them all together. Thousands and thousands of different cells are all put together. We carry out one experiment in a tube, and then we decode it by sequencing and asking what sequences are present. So it's a very simple experiment. Now, so it's pretty simple to switch over to doing DNA sequencing instead of hybridization. But if you look at these profiles then, for example, this is the growth rate with increasing concentration of the gene. This is the fitness profile based on how fast it grows. And what you're doing is taking each gene, and each gene is a spot, and ask where it falls. If it falls up here, then you probably have found the target for that drug. An example of that is TOR2, and its target, the drug target is the targets the TOR2. So it's relatively simple to figure out the targets. And you can do this in very high throughput. So you simply take these yeast and throw in a small molecule and ask what happens, and you get a profile, a growth profile. Then you do a different compound and do the same thing over and over again. Just keep going until you get a very large set of these profiles. And then you get a good bioinformatics student or a computer science and ask them to try to figure it out. Now, what happens with this is what genes are required for growth. This was one of the things that we started to do, and we thought we could try to do this with these drugs. And the pharmaceutical industry really said this will never work because you won't be able to get enough drugs to beat the curve. But what we already know is that 18% of the genes in yeast are essential. And we also know that another 33% of the genes show a slow growth if you delete them. So that's still a phenotype. So we're looking for what fraction of the genome can you generate a phenotype for? And this has been an old question because of this problem that I can delete a gene and I don't see a phenotype. So I want to know why are all these genes there? I can't figure that out unless I can get a phenotype. So this is the first principle of genetics. And then when we do all these different compounds, then we can now generate a phenotype in this first type of screen for 93% of the genome. What's left, in fact, about half of those are probably not even genes because they just fall out from the bioinformatics. And if we continue doing the screen, I'm sure we would get them. So that says that all of the genes of the yeast genome are important under some condition of growth. And that's what they're doing. The other issue is that the pharmaceutical industry has felt that there's a very limited amount of druggability. That most proteins can't be drugged. And because they can't be drugged, they're not trying. So we just did a separate experiment to look at really small molecule protein interaction. And we did a small screen to see how far we could get. And in this small screen we identified 55 gene products that we could find a drug for that was specific. And given the number of compounds that we screened, a large fraction, I think, of the genome we can find good drugs for, or molecular probes. I want to keep focusing on molecular probes because drugs are something that you use for humans and there can't be any bad cross reactions and all that. But you can use, biologically, you can use them as probes. You can take this and shut down the gene product and use it as a substitute for genetics. And it has a lot more power for doing a lot of biology than just immunity. So I think that the druggability is much larger. There's another experiment that is that another category of drugs that you could do are protein-protein interaction inhibitors. So there's a protein, two proteins interact, and if you can inhibit that interaction, you have a lot of possibilities. We wrote a grant to do that test. It was initially turned down because people reviewing the grant were from the pharmaceutical industry, and they said it would not work. We then went forward because of our grant situation and showed that yes, it does work. And in fact, you can find compounds of this nature. And now we're going to set up a much bigger screen looking for protein-protein interaction. We'll use our use system to do that. So I'm going to just show you one last thing. Because I think there's a lot of possibilities for developing new drugs. We have a new project to develop new drugs using synthetic biology and then with genetic selection. So one of the problems in doing some of this stuff is getting the drugs in and so forth. I think it'd be a lot better for our screens. Also, we have to make the compounds. Is if we could use the yeast to not only screen the drugs, but also make them. And then our limitation in terms of compounds is no longer there, and we can start screening millions, if not billions, of compounds. So we decided to do this with the polyketides made by the Streptomyces. If you don't know much about the polyketides, they're a very interesting class of chemical compounds, and I decided not to show you what they are because we're taking another lecture this time. But they're made by Streptomyces as two carbon build-up units. You put in two carbons, and then you may modify one of those carbons by putting an amino group or an alpha group on, and you add two more carbons. It's like fatty acid synthesis. And you build these compounds up, and then you do a ring closure or cross bridges. But it's all done by one big molecule. Every reaction is a separate domain. So these polyketide genes are multi-domain, each carrying out part of the chemistry. And it probably, it's not the same as the chronotoxins, but the principles are there. That is, there's something that can generate a lot of diversity. And so we're going to start out by doing DNA sequencing to massively sequence these genes to find out what are the collections of genes. And now we can then synthesize them to work in yeast. Problem with that, of course, is it's a lot of DNA synthesis. We built a new oligosynthesizer. This instrument does 3,456 oligos at a time. It costs us about 10 cents per 100 units. And that's going to pay our instrument to do synthetic biology. So we can synthesize the entire yeast genome for about $12,000. So then going back to this, when we get into the yeast, we're very good about making a gene function essential for yeast. And either that because it's similar to a yeast gene, or because we can set up some kind of complex reaction. And we can probably target many human genes, things like HIV, TB, and malaria, especially if you do protein-protein interaction. It's really trivial to do that. And so these modified yeast, which will be adapted to these, and it'll be adapted to do thousands and thousands of different assays simultaneously. We then put these synthetic molecules into the yeast, and then scramble them. So we use the yeast recombination system to evolve those new products. And this allows rapid evolution of the polypeptide. We are already built into here all of our targets. Then we'll make the drug in yeast. In addition to finding it in yeast, we'll simply manufacture it in yeast. And then the last step would be you make beer. And instead of purifying the drug, you simply drink it. Now, we thought this was a great idea to keep the cost down. And then my student discovered that 2,000 years ago, the Indians actually were making beer that had antibiotics in it. And so this idea has already been tested. So it might be a way to deliver drugs, especially things like anti-malarial, anti-TB, anti-HIV, drugs very inexpensively. You could use mixed populations to make mixed drug deliveries as well. And it could be all in the same beer. And a nice advantage of this is to make multiple combinations of those drugs, so that in fact you have different types of beers which have different sets of drugs, so that you generate the diversity there, so it will help you get rid of resistant selection. So I think there's a lot of opportunities for technology development, all the way from very sophisticated things like the nanofabrication to simple ideas like this that really just involve genetics and yeast. These ideas can be done almost by anyone. And so what the stimulation needs to be is to think about how to solve problems and what are the problems, and keep thinking about it. They don't happen overnight. You have to keep working on them. But if you do work on them, you'll come up with something, generally speaking. But thank you very much.
Are there any questions?
So again, one of the real world problems is that for sequencing or for detection or quantification, being able to deal with the presence of small numbers of viruses or bacteria that are in an environment that is a large volume and may be a difficult one, a sputum or an amniotic fluid or whatever. So how do you see dealing with that going forward? It's essentially a purification or a concentration problem.
Well, two ways. One, if it's already nucleic acid, then it's already mixed. Now you're trying to get the nucleic acid out. So the next postdoc of mine right now is developed an electrophoresis instrument that can take 55 gallon drums of liquid and concentrate all the nucleic acid down into a microliter. This is just by electrophoresis. It's a pulse field gel, and he just worked out that he did it theoretically that he could actually make DNA move to the center and out of a large volume. because this time electrophoresis works. Now, it's still mixed, and sometimes you don't, it's not DNA that you're looking for, it's a part of the virus, bacteria, as you said. So we've been working on a technology to solve that. Especially if you go into human, you have a lot of human cells, and you have a lot of human DNA. You can't see it. Even if sequencing is really cheap, you still can't see it, but it's way too little. So what we're doing is working with chemical engineers. I have a couple of students in chemical engineering. And what we're doing is taking all of that sample, let's say it's 10 mils, and we are then compartmentalizing. We're putting it into tiny vesicles. These vesicles are about, well, we can control the size, but depends on what you're going after. But a micron to 10 microns in cells. Depending, if you're going after a human cell, it'll be bigger, the bacterial cell is different. And so, in fact, we encapsulate all the sample. And now, but those, we can control the porosity of those vesicles. And so we can actually do DNA prep. And we can get rid of all the protein. So we can actually do the DNA preparation inside of the vesicles. And now we then sort the vesicles for something else. So we could put in, for example, if we want to get rid of the human cells, we would put a stain in that would stain the human DNA and throw those away. If we knew what we were going after, like a bacteria, there are sequences that are highly conserved in bacteria. The RNA polymerase subunit is highly conserved. You could go after that and then sort out all the vesicles that have any bacteria. The reason to do this is a lot of people are trying to sort out these complex environments where there's a mixed population of bacteria, but the bacteria won't grow, so you can't make a colony of it and have a pure culture of it. So they think they can sequence the bulk and assemble it, and I think that's complete garbage. These organisms are too closely related, but sometimes quite mutually exclusive. And so you will put together an organism that doesn't actually exist. And you'll miss a tremendous opportunity that you see some cases that certain species have certain combinations of genes that aren't found in other closely related species. And it's probably for functional reasons that that's there. And you need to preserve that information. So the hard part that we weren't sure we could do is done. So now a matter of turning the crank and getting that into some applications. So we're optimistic we can do that. So it'll be applied to bacterial, probably gut flora, so we can actually get the entire genome of the gut flora that will not grow. So we can compartmentalize their genome. But I think it has a lot of applications. But you're right, you spotted a really important problem that a lot of the genomicists are swept under the rug. And this is an example too. When you don't have an idea how to do it, you just pretend it doesn't exist and go forward. Well, that's a big mistake. You got to look at the problems and then solve them, or we'll get nowhere. So I appreciate that.
Any other questions?
So it seems like the drug companies have targets in search of drugs, and you have all these molecules, and you're going to marry them to all the possible targets in the organism. Is that the way I...
Yes. Well, what drug companies do is pick a target and then screen large numbers of compounds.
I'm sorry?
They pick one target and then go after
the drug companies.
Right.
Yeah. And you're doing it the opposite way.
Yeah,
and you could have this huge library
right.
of molecules that you can just pick off the shelf whenever...
Right. Use commercially available materials at the present time.
Right.
But that's a little... but now we're finding it's a little limited. The problem is that there are rules that the drug companies have decided are rules for what makes a drug. And so when people make these compounds, they follow those rules. Well, we've tested those rules with this system, because drug companies don't come close to screening the number of gene products that we do. The rules are wrong. If you want to get good, high probability of getting a good inhibitor, there's a whole set of different rules that really apply. And we've done enough to see that. They haven't. That's the problem. They've done a few targets. We've done thousands of targets. So you can't buy them. Well, they're limited. So that creates a problem. So I'm more optimistic because those rules are wrong. And so there's a much higher probability of getting a drug. than they would estimate.
So if we do it your way instead of the drug company way, how is it going to be marketed? Who's going to make a profit on this?
Well, if it's a real drug, it's a problem because you've got to do a clinical trial. So that's hundreds of millions of dollars. So it'll be the drug companies. Or we have to change how we do testing. I would be advocate for trying to come up with a creative way to do testing differently to get the cost down. Drug companies love it, but it's hundreds of millions of dollars. It keeps all the smart people out. Right? I mean, they would not change that. But it's a real problem for things which don't have a big market. Drug companies don't want to go after anything. It's going to make $1 billion a year. Right? And there's a lot of potential drugs that benefit lots and lots of people if it gets fit.
One question over there.
Hi, sir. Maybe a related question. I'd like to, well, I was just curious. Has that been, for example, the chemogenomic profiling of yeast, has that been ever used to sort of identify some possible mechanisms or possible side effects of a drug, for example?
No, absolutely. We've screened all of the FDA-approved drugs, and a lot of them have listed side effects, and we see off-targets and it doesn't matter whether the yeast doesn't have that gene product or not, right? We'll still see some kind of reaction. Almost all of them make sense, including some of the drugs that in fact they don't know the target. They just know the drug works. So these are accidental discoveries. And we can find targets, and those targets make sense in terms of, yes, that is likely what is the real target of the human drug. We don't pursue it. There's too many of those to really pursue. We've been trying to talk the drug companies into doing this themselves and following up on it, but they're not interested. But you're absolutely right. You can see a lot of. I've been arguing that it's so cheap to do this yeast stuff. They should do it. They should do it early on in their drug screen. In fact, the chemists like to figure out what compound to pick. I would argue, do all your hits, screen the yeast. If it hits DNA polymerase in yeast, it's going to hit DNA polymerase in mammalian cells too. Don't follow that one. That one should be dropped. You'll often get a ribosome effect, that it binds with the ribosomal proteins. It's going to inhibit protein synthesis at some level. Don't follow that one. And use that as your guide early on. And just screen everyone. It's not very expensive. I have not been successfully convincing to do that. Because some of the things that fall out are because of some common low binding constant, nevertheless side effects. And we should try to get rid of side effects, if it's all possible. But I think side effects are such a big problem because they don't really incorporate them early on in this movie. And they just try to get around them in the end.
Thank you, sir.
Yeah.
I actually have a question that's more educational. So, in a sense, you have a very special environment, your center, because you can bring together people of very different disciplines in a creative way. But I think for the future of universities, it's clear that it's desirable to be able to do this at a very, very much earlier stage. And I was just wondering what your thoughts were on, in fact, how to do that, because of course traditional disciplines try to maintain, sort of keep the students so that they're chemists or physicists or computer engineers, but from your talk, and I think increasingly, it's clear that the most creative students should, have a window on completely different disciplines, and so it's fine if you're at Stanford and you're very well educated at that point. But how do you get this cross-disciplinary interaction at an earlier stage? So for the benefit of people who think about these things at I'm wondering what your thoughts on that are, a creative way to do that.
Well, from my perspective, the concept of an academic institution, you take all the people that know the same thing, and you take them and you put them together to talk to one another. That sort of minimizes your ability to influence. And then you do that across all the different disciplines. And then the further away the discipline, the further away you put the building, right? So you might put some of the biological sciences together, but your engineering would be on the other side of the campus. So you set up an institution like this to sort of to minimize any. And so that's a mistake from the first principle, I think. So I think one of the key issues here too is the fact that it's like, you know, mining for gold. And the disciplines have been mining for gold for decades, if not centuries, in their discipline. So you carved out, we're going to look for gold here, and we're not going to go outside and look. And that's where everybody keeps looking for gold in the same place. And yet there's a lot of territory that's never been looked at. So I think one of the issues is that when you go into these new areas, it's not hard to find gold. No one's ever looked there before. So it's an opportunity. And it's an opportunity for people to try to do that. So I think it just requires some encouragement. Now, what Stanford has done, I don't think quite realizing that they did this, but they've allowed students to move from one department to another. Now, they did that with the idea that it would be biology. So I can be in biochemistry and I can decide I actually want to do genetics, so I'm going to move to genetics department. Well, that's not a very big move. But they sort of established the principle that students could move. And so what's happened for me is that an electrical engineering student comes to me and says, hey, I want to work for you. Well, I say sure. But there's no rule against it. But how am I going to train an electrical engineering student? So then we start to work and find an advisor. Well, we first of all have to figure out the problem. And I don't want the electrical engineering professor to muck that up. So the student knows enough electrical engineering, and then that student trains me, and I train that engineering student in biology, and we work on a problem for a while, and sometimes it takes a year or more. And once we figure it out, what we're going to do, because something that doesn't really matter, we're going to pick something, some type of fabrication, some kind of discipline, and that's going to be his thesis project. He's going to interface with the biology. Then he goes and gets an electrical engineering professor that's a specialist in that area. And that means also that that professor will be very happy to advise the student, because that's that professor's discipline and expertise. If we start too early, then it'll all be, no, you should be doing this project, you should be doing that project. So I think that's one way to do it, is just to allow that to happen, or even encourage it. And in most cases, universities, they would discourage it. Oh, no, you can't do that. That's not done. And so it's more of an accident that that's happening at Stanford. They kind of aren't aware that it's happening, and I don't talk about it too much. So I have many more students in electrical engineering and computer science and chemical engineering than I have in biology. I have only one student in biology.
So I think we'll close the symposium, and again,
this is 100-fold better than I imagined, this symposium. I think the rest of the audience agree, and it touches at the heart of how we should do research, or how we should reinvent research in the university and have an impact on education as well. Different kind of way of thinking of things, different worldview. I think it's very timely because we're going to launch the Philippine Genome Center, and our director, Dr. Menchit Padilla, is here, and I'm sure she'll agree that aside from looking at the different programs that we already have established and looking at bioinformatics, now we have to include other areas that contribute to the kind of genomics or genetics work that we would like to do in the country. Okay, but I think what our speakers have provided is this kind of a way of thinking of things in a creative way. And so at in my office, we're trying to reinvent the research process as well. And I was, just preparing the documents, but now I'm really tempted to rethink this process. And I think the first thing we should do is to try a little congress where we'll get the best and brightest minds among our young faculty, researchers, and students to think about the big problems, and then different disciplines together in one meeting, and identify priorities, and then perhaps make sure that the students, the most creative ones, get the right background or get to interact with a complementary discipline. I'm sure many of you have questions to our speakers, and at this point I'd like to thank them for such stimulating talks. And I think there are about 40 of us here. For those who have stayed on till the end, we have lunch for 40 persons. So I think we can all go for lunch, or maybe a little in excess of 40. So I hope that you spend a little time talking to our speakers after they take their meal, and ask them all the questions that you want. And maybe this is the start of something that we can do in the university that's different. Okay, so let's give them a big hand. There's one other talk that Dave Hilliard will give at UP Manila. Is it in PGH or in NIH? That's on Friday at 2 o'clock. Okay, so those of you who are interested in more of the molecular diagnostics for infectious diseases, go and catch Dave Hilliard in UP Manila. Okay, thank you.